Good afternoon or good evening, everybody. So hard act to follow, Oisín, but thank you very much for that extraordinary off-the-cuff talk. So obviously, so the title of my talk is roughly Virology of SARS-CoV-2, but I'm going to try and give a very quick global perspective of some of the molecular epidemiology, a local perspective, and just an insight into some of the COVID-19 related research activities that are happening in UCC that I'm involved in. I suppose just a bit of background, I have a PhD in immunology, which was obtained with Tommy McCarthy in UCC, and then I went to Canada for a few years looking at VDJ or immunoglobulin antigen binding at the University of Toronto in Canada. And came back then to Ireland to complete the setup of a molecular virology unit, which was on foot of the anti-iatrogenic infection on hepatitis C. And that morphed then into the Molecular Virology Diagnostic and Research Laboratory, of which I became director. And I suppose that led to a union then between immunology and virology. And I suppose I've been forging a pathway of immunology and virology kind of in a lonely room here in the clinical sciences building for years, waiting for something maybe like coronavirus to come along and give us an opportunity when virologists were seen as more than esoteric members of the research community within UCC. Never mind actually somebody looking at immunology and virology. So to that end, and to give a nod to the Nobel Prizes. The Nobel Prize this year was awarded for the discovery of hepatitis C, and it's nice to see that three gentlemen who were involved in the early clinical definition of non-A, non-B hepatitis, Harvey Alter, Michael Houghton, involved in the laboratory discovery in Chiron Corporation for the first molecularly discovered virus, hepatitis C in 1989, and Charles Rice, who was instrumental in the development of laboratory-based methods for the investigation of the life cycle of hepatitis C, and latterly the investigation of agents which were to evolve into the direct-acting antivirals, which have led to the curative treatments that we have for hepatitis C today. So just a nod to hepatitis C and the years that I've spent working on and continue to work on hepatitis C, and the union then with that and the immunovirology, which leads to a kind of a natural segue into the immunovirology that we're hoping to get going in UCC with COVID-19. So without further ado, I'll move on to the next slide. So just again, people are probably familiar with this, but just to give an idea as to the kind of the speed at which this virus spread around the world, and the, I suppose, impact with respect to the numbers of infected that have been released as of today. So it's ironic, myself and a colleague, John McSherry, were actually in Beijing in October at an immunology conference talking about, well, I was talking about immunovirology in the context of bacteriophages, and John was talking about his work on asthma and BALs in the context of immunology. And little did we realize that obviously in December, as we all know, in China, the first patient started to develop symptoms of a pneumonia type associated with a probable viral etiology, and we had the Chinese government then alerting WHO at the end of December. And then the rapid identification of a new coronavirus, SARS-CoV-2, or COVID-19 as it's subsequently called, within a week of the notification of the WHO of this potential new viral pathogen, just showing the, I suppose, the advances in molecular virology or molecular technologies for sequencing new pathogens, and how important it has become as a way to integrate new and emerging pathogens into the management of human conditions. And 4 days later, China recorded its first death, or notified its first death from COVID-19 on January the 11th, 2020. Rapid movement then throughout the globe. Thailand, 2 days later, reporting its first case. And then, as we know, China introduced stringent quarantine measures in Wuhan, limiting rail, air, and even movement of individuals outside accommodation and that. So that's just the whirlwind stop of what became relevant very quickly. And I suppose it became evident in Ireland that this was coming, or in Europe it was certainly coming. And especially with the notification on the 14th of January 2020 that WHO warned of the risk of possible human-to-human transmission of the new coronavirus. And on January 20th, USA announced its first confirmed case in a 30-year-old male returning from Yuan. And at this stage, we had moved on from Thailand, and we had cases in Japan and South Korea. And as we are kind of all very much aware of the stringent measures introduced in Japan and particularly South Korea, and its use of tracking technologies to bring their particular epidemic under control. And then in Ireland, in green here, on the 29th of February, kind of ironic in a leap year, to have the first case announced in Ireland involving a male returning from northern Italy. And then everybody was waiting for more or less the WHO notification that this was more than just an epidemic. It was in fact a pandemic, and that was announced on the 11th of March 2020. And it has to be said that, you know, new pathogen December, availability of some tests in early January, and then the availability of CE-IVD marked kits very quickly thereafter, and the ongoing development of tests, PCR considered a standard of merit, and then we have other assays which use non-amplification technologies, which are trying to speed up the detection of COVID-19 from samples that are more accessible than the nasopharyngeal swabs that are often used to swipe the cells at the back of the throat for detection of virus that has been spread. So just to give you a sense, I took this picture yesterday and I decided I'd look at the picture today and to see how the spread of this infection. So yesterday there were 38.1 million cases globally. There are over 500,000 more cases today, and the deaths have increased from a little over 1 million to actually 7,000 more than they were 36 hours ago globally. And now there's a bit of wriggle room on these figures, but it gives the sense that, you know, this virus is holding no hostages, and it is a clear and present danger to the human condition. You've probably seen some of this throughout the week, but just to refresh, it's an envelope virus, small standard size virus, which has a lipid coat impregnated with various proteins which are of significance with respect to binding to receptors to get into its target cell, and also some proteins there which interfere with the activity of interferons, in other words, interferon agonists. So the virus, if you like, comes preloaded not only with receptors to get into particular cells, but also with an arsenal of molecules which actually dampen down or interfere with the immune response that we would trigger when we detect a pathogen such as an RNA virus. It's a reasonably largish genome for an RNA virus. It's 30,000 nucleotides, which is 3 times bigger than Hep C, 10 times bigger than HIV. It has 10 genes, but there's lots of overlapping reading frames, and we get 29 proteins from these. And there are 4 proteins on the surface, the key one being the spike protein, which binds to its receptor called the ACE2 protein, which is on plenty of cells in the lungs, obviously, in the salivary glands and the small intestine. And so it's relatively prevalent throughout the body. It's a protein that seems to have been under rather little selection pressure through human evolution. And it seems that some of the susceptibility to coronaviruses actually comes from our ancient evolutionary cousins. Our Neanderthal cousins seem to have endowed us with a protein, the ACE2 receptor protein, which seems to have a very high affinity for spike protein on coronaviruses. And I suppose a bit like other RNA viruses, such as HIV, influenza, and hepatitis C, it does have a genome that undergoes mutations, but interesting enough, it has an RNA polymerase which actually has a proofreading capacity, which actually means that the genome is particularly stable. And even though there have been trillions and whatever is beyond trillions of replication cycles in individuals during an infection, the actual genome sequence of the backbone sequence has been remarkably stable, even in the context of a polymerase which has proofreading capacity. And these different genomes endowed with mutations, obviously we're referring to them now as strains, even though they're more kind of quasi-species or kind of subtle differences in the genome rather than strain differences, which, we don't yet know whether these kind of mutated or different genomes have slightly different natural histories, be that infection competencies, capacity to overtake the cell machinery, and once it's infected, our capacity to downregulate or modulate the immune response or impacting collateral damage by, we'll say, changing the clotting capacity of the normal kind of clotting mechanisms, factor V and fibrin. The main test, as I said, is a protocol which changes the RNA into DNA, and that DNA and then is amplified, and it's called PCR. And we're using fluorescence then to quantify the amount of virus. This has led to some discussions about the value of some of the RT-PCR assays with respect to its high false negative rate in the early stages of infection in the first couple of days. Some of the early papers indicated that it was quite high, 30, 40, 50% false negative. Depending on the, obviously that is dependent on the viral load and the lower limit of detection or lower limit of quantification if it's a quantitative assay for COVID-19. And then even up to day 7, day 8 post exposure, relatively lower levels of false negatives, but still nonetheless false negative rates of the order of about 10-ish, 15%. PCR does take a while. It doesn't take that long, but in the context of wanting to know results immediately, it takes a few hours. There are other tests which are, which take less, but it is a bit of give and take. The positive results and the quicker assays, assuming sensitivity and specificity is appropriate, you may lose some sensitivity with the quicker assays, which you can compensate for by a slightly longer, more classic assay with the RT-PCR. But the problem with the RT-PCR is everybody was doing it, and we ended up with reagent shortages and actually nearly a collapse of the testing environment in some countries as a result of chemical shortages, which were responsible for bursting open that envelope around the virus and getting access to the RNA, so that the enzymes that are used in RT-PCR could then do their work and give us a signal as to whether there was detectable levels of the virus or not. So I'm sure, by in the next couple of months, we will have assays that can be done at airports, at peripheral sites, pop-up sites, that will have a lower level of detectability that's acceptable, and rates of false positive and false negatives that within a workable range. So just to give you some sense of the mutations of this virus, on the image on the top right hand side, the one prior to March 1st, you'll see it's blue with some orange, and that's predominantly the viral type at that stage was an aspartic acid at a particular point in the spike protein, the main protein on the outside of the coronavirus. It's called D in the one letter amino acid nomenclature, and this changed, or it was popping up and down in the early days of the pandemic, but there was a big shift in March, April from the D614 to the glycine variant on the spike protein. And some evidence is suggesting that this is potentially more infectious. It's not terribly, it is robust, but there's little enough of that evidence around. So I think people are more interested in whether this leads to a differential disease outcome as opposed to being more infectious. And we await to see, further information on that. So all this genetic mapping has led to, a kind of an appreciation of the value of phylogenetic networks. It was very much used in the early days of iatrogenic and in hepatitis C, with the, trying to link individuals who received hepatitis C infected anti-D immunoglobulin phoresis incompatibility and the original inoculum, and to sequence and identify high degrees of similarity, and that was taken as kind of like fulfilling Koch's postulate that the two samples were related and that the infection was as a result of iatrogenic iatrogenic received hepatitis C. So much in the same way, the molecular epidemiology has interrogated the genome sequences, and what you get basically is this kind of spoken wheel and hub effect, where you can see the ancestral strains down the bottom right hand side of the screen coming from one of the putative sources of this zoonotic infection, bats, and then the evolution as it migrated from bats into humans, and the early transmission amongst humans, and then the outward global movement. And we can see this kind of color coding there according to countries. Obviously, this data was published in April, gathered at the first 3 months of the pandemic. And while China is still the epicenter and contained a lot of the dominant strains of the phylogenetic network, we can see that it was spreading out around the globe rather efficiently, and that there were notable synonymous or changes that resulted in amino acid changes, or just genetic changes that didn't result in amino acid changes that probably had effects on fitness. And this is a map of the genome sequence of COVID as revealing a global transmission network. And what it really affects is a map of airports. You know, we have, you know, plenty of spread around China before China was closed down, spread within Thailand locally, and then spread to the Americas and into Europe. So, you know, modern travel has in many ways a lot to answer for the spread of this particular infection. A bit like SARS-1, it spread to the few locations around the globe as a result of airline transmission, but as we all are aware, very effective containment brought that particular viral potential pandemic under control. And that leads me into the Irish kind of efforts to understand the coronavirus infection. And what was set up and funded through SFI was the Irish Coronavirus Sequencing Consortium, led by Professor Paul Cotter, who's head of food biosciences in Teagasc, from Moy here in Cork. And you can see there on the left-hand side a large collaborative working research group across the country, collecting and currently analyzing samples from individuals on an anonymized basis to understand the dynamics, the entry or the number of frequency of entries or likely frequency of entries within the Irish population, and where we can kind of map the second wave that's coming, and you know, and how we're going to learn with regard to kind of setting up phylogenetic and molecular virology analysis of these large outbreaks. So just to give you, put some pictures behind the individuals, we have Fiona Crispy there and John Kenny from Teagasc on the left-hand side, and then the eclectic mix of individuals pictured, some not, of the cohort of individuals who are striving in a kind of a multidisciplinary team approach, clinicians and researchers, to understand this. We're hoping to analyze about 2,500 samples across the country, and again, this is to preserve anonymity. No identifiable details are being collected. We're only collecting gender, age, and region or voting constituency. Kind of local electoral areas will be collected to try and assess the movement if possible, and that's dependent on the kind of sequence variation. And we're using a sequencing approach. It's nothing really strange. It's just kind of all brought under the umbrella of next generation sequencing. RNA is converted into the single-stranded DNA. That DNA then is amplified, and then we run it through a series of machines. One of them is only the size of a USB key, so it's amazing how small these sequencing devices have become. You get millions and millions and millions of sequencing reads. They're bioinformatically reassembled, and you end up getting, if you like, a series of genome sequences, which phylogenetically, when blasted against reference sequences, end up with this confusing looking tree. And what you end up then is, when you identify your Irish sequences, and the Irish sequences are one in the red oval, they're clustered matching sequences all around the globe. And what this tells us is that there were multiple entries of different sequences from various regions around the globe in Ireland. Again, I suppose nothing strange. Ireland wasn't shut down. Ireland was left very much open. And you know, hubs within Europe, London, Italy, and from China and the Americas back into Ireland. So this back and forth across the globe, bringing this kind of a real mixing environment for COVID-19 into the Irish population. So what the output of this is data collected pre-June, and there will be much more rich data set published sometime early next year, once when those 2,500 samples are analysed from the first wave, primarily March and April, with some samples in May and slightly fewer samples in June. And there will probably be a second iteration of this consortium looking at the second wave. So this is sequencing consortium, and just let me backtrack. A lot of that data and sample collection was thanks to the efforts of Killian de Gascoigne, Michael Carr, and Gabo Gonzalez, and Killian de Gascoigne is director of the National Virus Reference Lab. So he's kind of a busy enough individual these days. So in blue, just have highlighted kind of the Cork contribution to this. Colleagues John McSherry in UCC and Mike Prentice cross-appointed to Cork University Hospital and UCC, and colleagues in Teagasc Moore Park there, and the plethora of other colleagues. You can see this is a truly national effort with collaborations and involvement from the UK. We're not just doing molecular virology. What I have here is 10 projects that I'm involved in, and the diversity of projects that have grown out of COVID-19. And I suppose even in a pandemic, there are opportunities. So, you know, we're looking at a diverse range of aspects of COVID-19. We're looking at the neutralization potential of anti-COVID antibodies in healthcare workers who were infected with COVID-19. We're looking at saliva as a matrix, as a possibility for testing for COVID-19, because the nasopharyngeal swabs are actually quite uncomfortable. You know, if we could have something that was robustly validated as a matrix, then saliva would be, you know, usable for all ages. We're looking at the microbiome, how it changes the metabolome within individuals. We've a study looking at students who are coming in from abroad, and in the restricted movement period, looking at the prevalence of both antibodies. I'll show a few results from that in a few minutes, and whether they have detectable levels of the virus in saliva. Immune cell function, microdroplet capture of COVID-19, and we have a project on sewage. Sewage, believe it or not, is a very nice medium for detecting the presence of COVID-19, and it's sentinel by a couple of weeks for large outbreaks in urban populations, and is, you know, may become an important sentinel for undetected or unrecognized asymptomatic infections, maybe in smaller communities that have local sewage treatment plants or small treatment areas. So that's just a funded by SFI, involving collaborators from UCC, HSE, Cork University Hospital, and other hospitals, and the clinical research facility down in the Mercy Hospital. Just to give you a small nugget and insight into what we're doing with some of the antibody work, so joining the immunology and the virology together with colleagues in the clinical research facility, UCC, and Cork University Hospital. So where we were looking at the seroprevalence in healthcare workers of antibodies against the spike protein, that protein that I told you earlier on that is on the outside of this virus, and is one of the main diagnostic antibodies for looking at whether individuals have mounted an immune response, a humoral immune response, except in the fact that, of course, there can be T-cell immune responses as well. And we had what was a point of care test. So basically, these are like pregnancy tests. Technology, it can be done on the bedside, it can be done anywhere. It's a test result that requires very little technology. You really just need something to puncture the skin, and then apply it to the device there on the bottom right hand side. You apply a carrier liquid, and within 10 minutes you'll have a color development. And I've just shown two examples there. The one on the left hand side is an example of a sample that had no reactive antibodies, IgG, to the spike protein. And what we have there in red is the control line. And once when that's red in color, we know that we have a valid test. In other words, there was sufficient material or blood applied, and that if there was antibodies present subject to the threshold of these lateral flow devices, it would have been positive. And on the right-hand side, you can see that we have two bands, one C and one G. And so there, that the blood applied to that particular point of care test had antibodies which reacted against the spike protein on this point of care test, indicating that at that moment in time, they had antibodies against the spike protein. And what we found through looking at this is that not everybody holds on to these antibodies that are at a detectable level with these point of care tests. And we have some individuals who have been positive on their first point of care test, and sometime later then we have found them to be not detectable. It doesn't mean that they haven't got antibodies, it just means they're not detectable with respect to this particular lateral flow test. And the significance of this loss is unknown. We still don't know if these individuals will be less immune, and whether they'll be able to recall a memory response, be it a B-cell memory response or a T-cell memory response. And to that end, one of the questions is, part of the natural history of COVID infections is sequential infections, and whether it's possible to have an infection have an acute resolving infection, and then can one be exposed and can one have an established infection? Anyone who has had an infection, be it influenza or hepatitis B, and has had an acute resolved infection with either of those two infections, mount an immune response which for hepatitis B will be long lasting and will be neutralizing. For influenza, it will be short-lived, it will be neutralizing, and memory may be embedded or may not be embedded, and may last or may not last for a long time, depending on the individual immunogenetic parameters of the individual, the extent of the disease, the infection with influenza, and whether they were vaccinated or not prior to that. And so one of the leading questions with regard to, obviously, is we're all concerned about COVID-19, the sequential infection. Does it happen? Yes, it does. There are a few cases. The actual incidence is unknown because these are picked up, if you like, serendipitously, for the most part, in asymptomatic individuals. So there was a nice case study published in The Lancet just last week on genomic evidence for reinfection with SARS-CoV-2 in a case study. There are some, but I'll just give you some insights into this one. So this individual was a 25-year-old male who sore throat initially, headache and nausea at the first outbreak of COVID-19 in the USA on the 25th of March 2020. Funny enough, the first viremia reported was 24 days after infection. Most individuals, their viremia would be reduced to non-detectable levels at some stage before day 24, certainly maybe within the first 14 days post-exposure. However, they had a resolution of symptoms roughly a month from the onset of symptoms, and then through 2 different molecular amplification assays had non-detectable, they report negative, but I would tell you they're non-detectable levels of infection. So just for the moment, accepting that negative in this case means 0 and no level of COVID-19, the individual returned 2 days after their last real-time RT-PCR, classified as negative, with symptoms, and then tested a week later and found by RT-PCR to be positive. And then they didn't present data for the first infection, but the second one they presented data to suggest that they were IgM now detectable and IgG detectable. Familiar story, genetic analysis, and what I've highlighted here in the kind of pink oval is the U1 strain, the initial strain for the pandemic. And then the other 2, one in red was the specimen from time point 1 or time point A, and then the specimen from time point B. And that there was sufficient genetic divergence between them to still classify them on similar branches, but nonetheless to be genetically distinct enough, but still within the one clade 20C as they had described it. So sequential infection can happen. We know it happens. It's likely underestimated, especially if the first infection was asymptomatic and the second infection is asymptomatic, and you're in an environment where there's, we'll say, lack of contact tracing, so they're not picked up. And so anyway, so the cause of this variation, purely speculative at this stage, could be the inoculating titer, could be the variation in viral virulence, subtle differences in the genome structure of the infecting virus, which may or may not cause a second and sequential infection. And some individuals may exhibit what's known an antibody-dependent enhancement. In other words, their first infection may be quite mild, and then because they have antibodies against the virus, when they're exposed the second time, these antibodies latch onto the virus, and instead of helping clear the virus, they actually help with uptake of the virus. And you end up with, if you like, a greater multiplicity of infection. And this may, in some cases, lead to a more serious symptomatic course of the infection. So that's me finished with a kind of a whistle-stop tour of what's going on in Cork, what we've done and what we have yet to do and hope to achieve. So back over to the chair.
That's wonderful, Dr. Fanning. Thank you so much. It was incredibly informative. Now, if you don't mind me asking, would you like to take the questions now, or would you like to wait till the very end?
No, I'll take some now.
You'll take some now. So we have one question sent in from Mr. John McSharry. Could you ask Dr. Fanning, do you think, oh, forgive me, it's after Yes. So could you ask Dr. Fanning, do you think they should be doing antibody and antigen testing on students to provide a bio passport?
Very interesting question, John. Thank you very much for that. Well, let me parse that answer into two things. The whole issue of passports is contentious. Let me divide the passports further into antibody passports and then antigen passports. Antibody passports are contentious from the point of view of that the data isn't robust enough to say that antibodies will give you protection against subsequent infections. Infections that establish and become symptomatic. Anyone who has mounted an immune response, in order to trigger the memory response, must in some level be de facto reinfected, but then we manage to control it. So antibody passports based on antibody seropositivity, you'd want to have, if that was to be a case, a WHO nearly approved world standard, so that everybody would be working off the one hymn sheet. And if I presented my passport in Spain, or got an antibody passport presented to me from Nicaragua, that I would have the same confidence and I would know the assay. The difficulty with it is that the time frame from point of passport, we'll say approval to point of transport, if they weren't really tight, then the validity of the passport would be weakened. I'm not coming down on whether there should be antibody passports or not. I'm just saying the validity of it will weaken. Antigens and antigen passports. Well, antigens are more relevant, as John will know, in the early stages of infections, and detection of antigens and roll out of them, be it with lateral flow tests like I showed for the point of care tests, is a matter that's under, if you like, urgent investigation across the globe. And we saw there yesterday, I think, was it that from one of the colleges in Cork, one of the secondary schools in Cork, that they were engaging in a study, a research study, and the utility of antigen testing. So students, be they secondary school or university students, testing for antigens has a place. If we can come up with an assay that has an acceptable robustness and an understanding of the caveats associated with rapid antigen tests, or the rapid tests in general, then they have a place. So long as the false positive rate wasn't too high, I think they have a place in triaging and perhaps having a screening program that would run in parallel to that of the national screening program for individuals who will be reporting in through GPs or their caregivers, whoever that may be. The testing of antigens in general at airports, it depends again on the robustness. I think if it wasn't for the fact that we could be competing for reagents for the national testing program, then I think rollout testing at airports has a place. It's not the only thing, and a one-time point test doesn't give you all that you might want from it, because of the rates of false negatives depending on when they were infected. So that's a roundabout way of saying in some places yes, in some places no, depending on the robustness of the assay in question. I think we have a duty to explore these rapid tests. They do have a place perhaps in preserving hospital capacity, where, as Oisin said, hospital capacity, and that means laboratory capacity, is going to be under serious pressure, especially if we end up in a situation where, like we were, Matt, where we're all competing for reagents to actually do RT-PCR on these samples.
Perfect, thank you so much. Now, our next question comes in from Dr. Jerry Killeen. So he's wondering, what are our chances of a vaccine conferring high levels of long-lasting protection?
Okay, so in order to answer that question, you must have some sense and understanding of the differential biology between receiving a vaccine, eliciting an immune response from a vaccine, and having an immune response from a full life cycle competent virus. So the life cycle competent virus, COVID-19, that people are being infected with, has many proteins which downregulate immune response. Even on the surface of it, the M protein downregulates as an interferon agonist. So the competency of a host to reply to the threat of a viral pathogen in the context of a full competent genome is different to that of a single protein, or if it's an adenovirus backbone or an mRNA, which is going to be converted into protein, is very different. They're not equivalent. From our understanding of the limited natural history that we have of COVID-19, antibodies in some individuals disappear rather quickly. Whether that means they disappear below detectable levels. Whether that means they disappear to a point where they're no longer effective and there's no memory recall, we don't have enough data. And so if you took it that those disappearance of antibodies fall to a point that's too low and there's no memory recall because of interference in the embedding of memory by the full genome proteins, then vaccine has a place even in those that have probably been exposed. Vaccines, you're only getting a single protein, probably with an adjuvant, and to spare the amount of antigen or mRNA that needs to be used to make it more widely distributable. I think it is probable that we will get certainly longer than many individuals experience with their natural infection antibody levels than with the vaccine, or maybe even with the mRNA. Whether they will, what Jerry means by long lasting, well, anything longer than in the context of COVID-19 than 3 to 5 years will be, like 3 to 5 years will be probably, listening to what, you know, Oisin and others have said, Dr. Tetris from WHO, if we got a vaccine that even gave us antibody protection for even 2 to 3 years, would provide some populations with more protection, would help protect the health service, would maybe, you know, assuming that this would reduce symptomatic infection, would still be of value. I don't think we need to necessarily have a vaccine that's neutralizing and sterilizing like the hepatitis B vaccine. I think we need a vaccine that supports and probably protects the health systems that we have. So that's a kind of a way of, I do think vaccine is different, and people shouldn't really be comparing the antibody responses that we're getting from a natural infection to that we're going to get with a COVID-19 vaccine, which is a protein subunit or an mRNA.
Perfect. Thank you so much for shedding light on that. And our final question is there anything that we can learn from sequencing the catalogue of viruses endemic in bats to prevent or counter future outbreaks?
Okay, well, I can use that question as an opportunity to make a play for the biosafety level 3 lab that we're trying to develop a plan for its sustainability beyond COVID. So this asks a question beyond COVID. I mean, we're all focused on COVID, but you know, 2018, the WHO produced a document which basically described what COVID-19 was going to be. The pandemic that challenged health systems, that challenged vulnerable groups and populations and put health systems under threat, for which there was no pharmaceutical or immediately available therapeutic agent or vaccine that we could borrow from another pathogen. And we definitely need to be looking at the endemic viruses in a whole range of animals that humans come in contact. The next pandemic could come from a Holstein Friesian out in Coachford. It doesn't necessarily have to, you know, come from, you know, a market, a putative market, if that was the source of this. Remember, HIV came from, you know, probably, you know, eating or coming in contact with bushmeat in, you know, the Democratic of Congo around with the 1890s, 1900s, to give or take about 10, 20 years on either of that. And so these things are happening all the time. It's just that we have paid attention to this COVID-19 one because it has caused such economic and such, if you like, health misery across the globe. So I do think we need to be cataloging not just bats. You know, we need this, you know, to misquote from Lord of the Rings, the time of virology has come in immunology. It's time that the world paid attention to these things. And if we don't, we're going to end up having, you know, subspecializations within respiratory medicine, looking at particular nuances of particular viral infections that suddenly throw off clots, and we end up with strokes in 27-year-olds, because, you know, we haven't invested. You know, this back to Oisin's point. We got the warning signs. We didn't pay attention to them. There are 5 viral pathogens on the WHO potential pandemic list still. And yet, you know, it would be very difficult to get research funding to start cataloging the known and unknown viruses in a whole range of animal, aquatic, plant life, you know, that could potentially cross over and be a zoonotic infection, which, you know, there's no reason to say that the next pandemic isn't actually already beginning now. They don't have to be sequential. Mother Nature isn't that kind.
Perfect, thank you. And I suppose the last question, if you wouldn't mind, is actually from myself. And listening to your presentation, I wanted to ask you if you think that the implementations that were put in place for the first SARS, so the SARS-CoV-1 virus, would that ever be possible to recreate now?
The answer to that is going to hold me a hostage to fortune, because no matter what way I answer that, we're going to have, maybe I'll be on Jerry's side on one, Jerry Colleen's on one side on one day, and I'll be on the more, I've had enough, my mental wealth can't take anymore, there's nothing left in the bank, and I just want out. Do I think, if you're referring to Ireland locally, I think it's going to be difficult. This is a personal opinion. I think it's going to be difficult to have a complete lockdown. I think we probably missed our opportunity earlier on. And it's not that we can't, but I think the collateral damages to mental health and other health, I know they're easy to say, yes, we're all very much aware of that, and there's a pandemic going on. But I think it's more nuanced than just saying, you know, will a lockdown? There are too many competing imperatives to give a very simple answer to that question. So basically, I can't really answer your question. There's too many competing imperatives.
That's perfect. Thank you so much for considering them all. And of course, you're completely correct in saying that a complete lockdown, whether it was back in March or whether it is going to happen again, it's going to have detrimental effects on other aspects of life, not necessarily just healthcare. But thank you very much for all that. It was very, very, very interesting. And it's the type of information that isn't, unfortunately, available to everyone from the get-go. So thank you so much for sharing. That was absolutely brilliant.
Just to say, for anyone watching this video in the future, thank you for listening.